I'm Scientist Magilaga, and today I'm going to tell you the process of protein synthesis. Protein synthesis is the process by which individual cells construct proteins. Both deoxyribonucleic acid or DNA and all types of ribonucleic acid or RNA are involved in this process. Enzymes in the cell's nucleus begin the process of protein synthesis by unwinding the needed section of DNA so that RNA can be made. The RNA forms as a copy on one side of the DNA strand and is sent to the other areas of the cell to aid in the bringing together of different amino acids that form proteins. Protein synthesis is so called because proteins are synthesized through mechanical and chemical processes in the cell. Once the strand of RNA has been made in the nucleus, it is called messenger RNA. The messenger RNA exits the nucleus through tiny openings called nuclear pores and moves into the larger area of the cell, known as the cytoplasm. Once it exits the nucleus, the messenger RNA is drawn toward the structure known as a ribosome, which serves as the cell workstation for protein synthesis. At this point, only one subunit of the ribosome is present. As the messenger RNA binds to the ribosome subunit, it triggers the approach of another strand of RNA called the transfer RNA. The transfer RNA strand looks for the proper place to bind to the messenger RNA, and when it finds it, it attaches to the messenger RNA while holding an amino acid on one end. When this occurs, the other subunit of the ribosome arrives to form a complete structure. As the ribosome surrounds the strands of RNA, another strand of transfer RNA approaches. This strand is carrying another amino acid and is different from the first. Again, the transfer RNA looks for the proper place to bind to the RNA. When the second strand of transfer RNA is in place with its amino acid, the two amino acids bind together with the help from the ribosome, as well as cellular energy in the form of adenosine triphosphate. This sequence repeats itself, and the chain of amino acid grows longer. When the amino acids have all been placed in the correct sequence, the chain folds into a three-dimensional shape. When this has occurred, the protein has completed. Once the protein has been successfully made, the two subunits of the ribosome separate to be joined together for later use. The process of protein synthesis takes place in numerous ribosomes throughout the cell. A cell which is operating efficiently can synthesize hundreds of proteins every second.
Protein synthesis. Transcription factors with the help of ATP decodes the DNA strands to form messenger RNA. Ribosomes wait until the messenger RNA exits the nucleus and begins transcription. In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of 30S ribosomal subunit, methionine RNA, and mRNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. One is called the peptidal or P site, and the other is called the acceptor or A site. There is also a third site called the exit or E site, where the RNAs are released. The initiating the RNA carrying methionine binds with the P site. A tRNA that recognizes the next codon and carries the second amino acid, then moves into the A site. The ribosomes now advances a distance of one codon. The methionine carried by the tRNA in the P site is then joined to the amino acid carried by the tRNA that just entered the A site by a peptide bond. The tRNA that carried the met is released at the E site. A tRNA carrying the next amino acid now moves into the A site where the anticodon on the tRNA matches the codon of the mRNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the tRNA in the P site are transferred to the new amino acid, and the second tRNA is released from the E site.
The ribosomes continues to move along the mRNA and new amino acids are added to the growing polypeptide chain.
Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding tRNA. The ribosomes disassociates into the 30S and 50S subunits, and the mRNA and protein are released.
Stereo, it beats for you, so listen close. Hear my thoughts in every note. Make me your radio and turn me up when you feel low. This melody was meant for you, so sing along to my stereo.
You class heroes, baby. If I was just another dusty record on the shelf, would you blow me off and play me like everybody else? If I asked you to scratch my back, could you manage that? Like if we had chicken travy, I can handle that. Furthermore, I apologize for any skipping tracks. It's just the last girl that played me left a couple cracks. I used to, now I'm over that. Cause holding grudges over love is ancient artifacts. If I could only find a note to make you understand, I sing it softly in your ear and grab you by the hands. Keep me stuck inside your head like your favorite tune, and know my heart's a stereo, the only place for you.
My heart's a stereo, it beats for you, so listen close. Hear my thoughts in every note.
Come on.
Make me your radio and turn me up when you feel low. This melody was meant for you, so sing along to my stereo.
Let's go! If I was an old school 50-pound boombox, would you hold me on your shoulder wherever you walk? Would you turn my volume up in front of the cops and crank it higher every time they told you to stop? And all I ask is that you don't get mad at me when you have to purchase Mad D batteries. Appreciate every mixtape your friends make. You never know, we come and go like on the interstate. I think I finally found a note to make you understand. If you can hit it, sing along.
Monday, party. Tuesday, party. Wednesday, party. Okay. Thursday, party. Friday, party. Weekend, 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 party. Wow. I'm telling to you, mama, there is no need for a drama. But I don't wanna study, I just wanna party. I'm a needy man. You know what a needy is? Don't wait for me awake. I'm coming home late. Maybe at 5, maybe at 6. Look at me, mom. I'm dressing like I just wanna party.